Okay, so I'll be the odd one out so far as a computational biologist. So nobody's perfect, as you know. So what does it matter? It matters because, typically computational biologists, we work on lots of different things. So instead of telling you one big story, I will tell you lots of short stories. That's one. And we are looking for broad patterns as opposed to specific processes, specific genes, so on and so forth. And so we detect stuff that typical experimental biologists may not detect, but then we can miss interesting trees while we are looking for the forest, and hence the need for working together. And as Usha said, don't be afraid to get into a new thing. So my story is from theoretical computer science to algorithms for biology to thinking about transcriptional regulation. And 6 years ago, I moved to NCI. I had no choice but to work on cancer, which I had never worked on before. So whatever you hear today, if you have any feedback, comments, I'm always here to stand corrected and learn, okay? So as I said, instead of one big story, I have few short stories. So we'll do it that way. Instead of doing in 2 parts, I have 5 stories. If I get through 3 of them, good enough, okay? And I don't know what the background of people here are. I'm guessing not many of you work with computational biology at any great level. So I will go slow, but stop me anytime during the talk for clarification. Okay? If I don't go through all my stories, that's totally fine. I can go through only 2 stories or 3 stories, doesn't matter. Okay, so general theme of what we do is transcriptional regulation. And so I'll get to the first thing. And those of you who are at my IISc talk, some of these stories are common, so you can check your emails or something. So first things first, if you look at any given gene's expression across clonal cells, you will see a variability of expression. If you see the same gene's expression in a cell across time, you'll again see variability. And in the second case, it's the identical promoter in front of 2 genes, and they are completely uncorrelated expression. Okay? So there is an intrinsic noise associated with gene expression. Some of you may be thinking, but it might be cell cycle phase. Obviously, with the cell cycle phase, you'll have different gene expression. But people have, of course, thought about it. And all these statements about gene expression stochasticity is above and beyond what you would see just because of cell phase, cycle phase. Okay? Now, if you think about this, like single gene has stochastic expression. And if you put that together with a gene regulatory network, what you get is cellular heterogeneity. And I'm illustrating that with a simple example here, the toy example with 2 genes only, and they have a specific regulatory relationship among them. And even though A and B by themselves could have that bell curve of expression, because of this relationship, all they can have is these 3 different expression combinations. Okay? Either both can be medium or one can be high, not the other or other way around. So this is what we consider as cell state or transcriptional state of a cell. So you can extrapolate this from 2 genes to 20,000 genes with a very well-defined regulatory relationship among genes. And what you get is this. So in any single cell data, the clusters you see are basically transcriptional states. Some of them are called cell types. You got epithelial cells, you got T cells, you got fibroblasts, so on. And some are within epithelial cells, you may have, epithelial or mesenchymal state. Among T cells, you may have exhausted T cells, so on and so forth. These are states and types. It's just a matter of degree, but ultimately these are our transcriptional states. Okay, so, but this random stochasticity has real consequence. So I'll just give you 2 examples. There are many, many out there. First example comes from mouse. This is a specific stem cell antigen gene, which shows this kind of bell curve expression. But the thing is, if you just sort out the cells with low SCA and let them grow, they ultimately regain the parental distribution, and likewise with the high and the medium. But the consequence, the physical consequence is when you induce differentiation, either to myeloid or erythroid, these different classes of cells have different efficiency to become myeloid or erythroid. So you can imagine if you mess around with the distribution tiny bit during development, you will have, that will affect the composition of these two types of lineages. Second example comes from, this is from development. This comes from cancer therapy resistance, melanoma, vemurafenib treatment. These are clonal cells. And just by chance, some cells will resist the drug. Okay? And these cells happen to have just accidentally high expression of AXL and other resistance genes. So these are called AXL jackpot cells. So they resist the immediate death, but at the same time, those specific cells undergo some epigenetic reprogramming, and that resistance becomes permanent later. So just to say that these stochastic expression has real consequences during development and disease and resistance. So when I came to NCI, we were just openly thinking about what to work on. And since my interest is in transcriptional regulation, every other day there will be some paper in some top journal saying we did single cell analysis and we found heterogeneity. And I used to joke back in those days saying that I would love to see a paper where the title says, we profiled 10,000 cells and we saw no heterogeneity. That would be really cool paper, but that never happened. So it's there everywhere, no matter where you look, it's there. And various heterogeneous cell states were being associated with different clinical outcome at that point, right? You have this cancer type, we did the profiling, and these specific patients with this specific cell state have worse prognosis or better prognosis, whatever. And our question was, what's so special about cancer, right? Why wouldn't this happen in normal tissues? So that was the thought experiment. So that's the question. Can cellular states in healthy tissue foreshadow malignant transformation? Malignant transformation just as a stand-in for something we are looking for when we are searching for heterogeneity in healthy tissue, right? Because what else will we look for? So this is the hypothesis. Like I said, we also think about hypotheses, but our hypotheses are about patterns, not about specific genes, specific phenomena, processes. So our hypothesis was, owing to intrinsic transcriptional heterogeneity, a subpopulation of cells in healthy tissue, that's a keyword, are primed for malignant transformation. Okay? How do we go about testing this hypothesis? And this work is done by Vishaka, who was at NCBS a while ago. So this is, in a nutshell, conceptually, this is what we are doing. So let's say we have some healthy tissue, which has clearly non-malignant cells, two different kinds, and we have malignant cells here. So what we are looking for in this homogeneous clusters of non-malignant cells is two things. One, do they have some cells which have just gone too far off? Okay, so we look for kind of skewness in the distribution of the distance of the cell from the center of that cluster. Okay, so just stop me if anything I'm saying doesn't make any sense. Okay, so and the second thing is, if we do identify those kind of high skewness, then we ask the cells that have gone too far from the center, where are they going? Are they going toward malignancy, right? So we are doing all this in the principal component space of single cells, right? So we applied these two statistical tests to every single cluster in a large PDAC pancreatic ductal adenocarcinoma data set, which was the only one available back then. Lucky for us, because it works like magic, this particular hypothesis in this data set. 57,000 cells, 35 patients, 24 malignancy, and 11 patients without any PDAC, some other pancreatic complications. Okay, right. So we applied this to every single cell type, and only pancreatic acinar cells passed these two tests. Ductal passed the first test, but not the second. Why I'm telling you all this? So PDAC, the name is pancreatic ductal adenocarcinoma. So there is an implicit assumption in that name that it comes from ductal cells in pancreas. But it's just an assumption, okay? It could be coming from acinar. It could be coming from acinar turning into ductal, then becoming malignant. So that is not resolved, and yet the name contains that term, ductal. So it's significant for us to find that our test only SNR passes our test and not the ductal. All right. So we found these. Now, for next 5 or 6 slides, I will tell you a whole bunch of biological facts about these edge cells. We call them edge cells. Okay. How do we biologically validate that they mean something, they have some function? So first thing, we looked at what is upregulated in these edge cells compared to the non-edge cells, and what are those genes. So in a nutshell, these SNR edge cells upregulate markers of embryonic pancreatic progenitors, SNR dedifferentiation markers, and SNR ductal metaplasia genes. And they downregulate markers of SNR cell identity. So they became a little bit dedifferentiated. They're not a typical SNR cells. By definition, they're veering away from the center, but in what way, right? So they're like dedifferentiated, and they're showing all these other markers, which are summarized here. And we specifically made some gene signature for multipotent progenitor for all these cells in pancreas, and showed that the edge cells specifically overexpress these and compared to non-edge, and this is true only for SNR and not for ductal for both of these signatures. Next thing we did was this Masco et al. Paper had collected lots and lots of lung tissues and staged them from completely healthy to a pre-malignant stage. Okay, they wanted to know the progression towards malignancy, what processes are involved. But this was not a time course data. Obviously, this is from real patients. It's staged, and they did that, and they put them in a line here, and they asked, okay, what are the processes or genes that are monotonically going up or down as it gets closer to pre-malignancy from completely healthy, right? So they found all these standard cancer hallmarks on this row that checked out. They go monotonically up. So we did same thing. Remember, these are single cell data. This is pancreatic ductal adenocarcinoma. This is lung bulk data. Huge differences. But we also lined up our cells going from the center toward the edge and divided them into few windows based on the distance. And again, asked, what are the processes that are going up monotonically? Okay. As you can see, it lines up so beautifully. The same types of things are happening in these cells that was observed in this staged patient data in lung. Next, so we have this signature now, the genes that are up in edge cells. So we asked, okay, are these prognostic in TCGA, which is totally another independent data set, nothing to do with our data set that we derived these signatures from. It shines compared to all other signatures we tried. This SNR edge up cells have the highest hazard ratio. But SNR edge down cells don't show a negative hazard ratio. So we just focus on this edge up. Yes? How would we prove that? That's an excellent question. Whether they were like this embryonal rest hypothesis you're talking about. Yeah, no, so stem cell, the notion of stem cell is extremely controversial in pancreas. So we can rule that out. That's not the thing.
Them at a particular spot. So if they're frozen at some point, they show up in your one point.
There is still,
yeah. If I understood right.
Yeah. Let me think about it. How we can rule that out? Nobody asked that question before. Because embryonal rest theory was kind of, I thought it was out, but that's a crazy idea. Let's think about it. Okay. Yeah. Oh, right. Yeah, right. Yeah. So like I said, so 11 of those 35 patients have no cancer, right? And our no detectable cancer, right, for sure. So that's one point. This phenomenon I'm talking about, we do it in a combined data. We also do it in a patient by patient basis, and it's robust. That's number 2. Number 3, we rule out using this infer CNV that they don't have any weird, chromosomal abnormality. So we did a bunch of things to make sure we are talking about, as far as we can tell, these are not malignant cells. And clearly, in the PC plot, they sit here. Malignant cells are here. They're not like getting there. They're completely in the healthy cluster. Oh, this is 2019. Back then, there was no spatial business at all, just bulk. Yeah. Crunch it up and sequence it kind of thing. Yeah. Right. Okay. Then we went to yet another single cell, healthy, completely healthy, nothing to do with any kind of cancer. Actually, one of them is from Van Uttanaran's lab, one of these data sets. What is this? This is just healthy pancreatic acinar cells, but sequenced using SmartSeq, not 10x. So the difference between those two is 10x is 3', and you have a little coverage of the gene. SmartSeq covers the whole gene, so it can be used to call mutations to some extent. Not perfect, but something in single cell level. So other good thing about this data set was there are multiple donors and their ages were known. So we simply asked, do the fraction of edge cells have anything to do with the age of the donor? Okay. So in these two different data sets, we see some correlation between age and the fraction of edge cells. And then the question comes, is clonality underlying these edge cells, right? So of course, that's why this notion of SmartSeq data set becomes significant, because then we can do mutation calling. So we did mutation calls. First of all, we looked at the entire collection of oncogenes and checked that their mutation, to the extent possible, there was zero. There was no mutation in any of the oncogenes. That's number one. Number two, overall, this is edge and non-edge cells. Overall, we see, it's not clear in this picture, but overall, there is a slightly higher mutation rate in edge compared to non-edge, okay? Now, it's known that when the cells are stressed, there is a mutation rate is increased. So that's not totally surprising. But the main worry we were having was, is there clonality? So that's why we are looking for the frequency of individual mutations across cells. If these cells were a result of clonal mutation, then we would see a blip somewhere here, right? Some mutation which is present in lots of cells. But that's not the case. Yeah, that's not the case. Non-edge, edge, they have very similar distribution of these frequencies. So we ruled, I think we have ruled that out. Because the community is stuck with genetics. There cannot be anything unless there is a mutation. Yes, exactly. It's still an uphill battle talking about transcriptional only based heterogeneity and, Okay, next thing we did was pancreatitis is a pancreatic condition which increases your chance of getting PDAC by X-fold. Okay? And there is a lot of research on pancreatitis. So we looked at a whole bunch of pancreatitis data and asked whether our edge signature is higher in the pancreatitis versus control. Okay? So lots of data sets. In every single data set, we see the signal, okay? Only one of them is not significant. And this is interesting. This was a time course data. And as the time goes by, you see a higher and higher greater signal here. So that's nice. This is a very interesting data set from Scott Lowe's lab, which is there they wanted to check the synergy between pancreatic injury and KRAS G12D mutation, whether they interact with each other, right? So they have mice with only chemical injury, mice with only KRAS G12D mutation, and mice where the mutation happens in the background of injury. Okay? And in that paper, the conclusion was, yes, there is a strong synergy because mice without the injury have some efficiency to malignant transformation, but with injury, that goes X-fold. Okay? And that's exactly what we see. So the KI is KRAS mutation plus injury. We see the greatest differential in that compared to these two. So now the last slide on this story, which is crazy, and we haven't done anything about it. So this Scott Lowe's data was in mouse. Okay. Yeah. Yes. Well, I mean, this is a time course, like 8 hours, 24 or 48 hours. So that's the time course in which we can see, clearly detect the difference. Yeah. Exactly. There are lots of arguments against it. The timing of everything is one big strike against the genetic, but we have to keep repeating these things. That's all. You're right. Yeah. Single cell data, telomere length, I don't know, but yeah. But yeah, that requires some thoughts. It's like, yeah. So KRAS G12D mouse versus the wild type, of course, differential gene expression analysis that they had done, right? And so that's on x-axis of all the genes, it's not selected genes. Y-axis is our differential expression of edge versus non-edge in human, in healthy pancreas. And this is kind of correlation you never ever see in bioinformatics. Okay, and all these genes check out. So what this is telling me is that what we are calling edge cells is like a phenocopy of KRAS G12D mutation without the KRAS G12D mutation, right? And it's kind of like two hit kind of thing. So injured tissue already has one hit. And on top of that, if you put the KRAS G12D, it's like two hit, and then the efficiency of malignant transformation is much greater. That's my thought process. I don't know. I have no proof. So this is really cool. And of course, we made a machine learning model of phenocopy and independent data set. We could predict which samples have KRAS G12D mutation just based on the transcriptome. So it's a real thing. Okay, so quick short story. The main takeaway is, so healthy pancreas and others, we had tried for 3 other tissues in the paper, express progenitor and SNR ductal metaplasia markers and oncogenic processes. Their proportion increases with age, and they transcriptionally mirror oncogenic KRAS G12D mutation, are potentially primed for malignant transformation. That's the take home. Yes, yes, I said that, I didn't show it. That's a very good question. I mean, we dealt with cycling issue very explicitly. We literally removed any cell that showed cycling signals. Acinar cells are long-lived non-cycling cells in homeostasis. So there are a lot of reasons for us not to worry about cycling business.
Sridhar, if there is any way to isolate these edge cells and inoculate in nude mice.
That's exactly what we have been trying to do since we published this paper. Just finding the right collaborators, clinicians who can do this hasn't worked for me. Yeah. Right. The ideal data set would be, pancreatitis patients, because those are chock full of these cells. Facial thing, right. And ultimately, we would like to manipulate this. Is there a way to manipulate edge cells to become, but at the same time, you don't want to start manipulating stuff that has evolved in nature, right? So who is to say that these things don't exist there for a reason, right? To deal with unforeseen stresses. So it's a double-edged sword. So you have cells in any population, you want to have some set of people who are ready to fight, to do something different. And that's what these cells are. And when you have these cells, they all can cause problem also, become terrorists sometimes, right? So
possible to make these cells? Is it stochastic? Like out of
that's a very tricky question. Yes, in our mind, these are stochastic. But the fact that they have slightly higher mutation rate somehow makes it hard to think, how can they go back to becoming non-edge, right? So we are imagining a situation where you have two types of cells, they go back and forth, but they cannot freely go back and forth if these guys have higher mutation rate. I don't know. All those questions are open questions. And again, for that question, you have to do lineage tracing, very precise lineage tracing to and check the state of the cell. We didn't look at our metabolic genes. Yeah, we didn't look at that. Sorry.
Sridhar, one more question.
Yeah,
Sorry.
of course.
So when you showed those graphs where you got this separation between survival, I guess, no, you mentioned that it associates with survival, right?
Yeah.
Yeah, you're right. But how do you exactly do this? Because if you have a patient, that means that person already has cancer.
That's true.
So you can't detect edge cells there, right?
That's true. So this one we did in the adjacent normal
I see.
of the patients.
I see.
That's a key point. Yeah. Okay, so good. Story number 2, we are ready to go. Yeah. Okay, wonderful. So second story is sort of, from thought, experiment perspective is kind of similar. So here the thing was, we know about cancer drug resistance. It's a thing, always is a thing, right? There are two kinds, genetic and epigenetic, and this is a beautiful review from Chris Marine that lays it out. In both cases, there is some analogy. In both cases, you have the concept of pre-existing resistant cells, pre-existing mutations, and mutations that are induced or selected upon treatment. Same thing with epigenetic resistance. You may have some primed cells that already are resistant, or sister cells, and you may have some plastic cells that become resistant upon treatment. Again, this general concept. So the thought experiment was as follows. Again, what's special about cancer cells, right? That was a question in the first project, same question here. What's special about resistance, right? So what is resistance? I mean, cell is trying to survive. That's what it's trying to do, nothing special, right? Cancers don't invent anything new. They just use whatever is available through evolution and just use it at the wrong time, wrong place, okay? So if a cell is stressed, it wants to survive, it has some enemies in the environment. There are only few things it can do. I mean, what can it do? It can pretend to die, like dead, like, whatever, quiescence, or it can fight, secrete some toxins. It can run away and migrate. What are the options? Or it can die if it doesn't do any of these things. Or it can recruit the microenvironment to protect it, maybe ECM around it or whatever, something like that. So these things have to be inherent properties of cells. Nothing special about cancer cells being treated by whatever drug. If that's the case, then shouldn't we see these, what we call resistant, long-term resistance signatures, shouldn't we see it immediately after drug treatment? That was the question. To what extent we see that? And not only that, if we see that a lot of this is happening early on, right after the treatment, to what extent all this stuff is conserved across evolution? If you mess with bacteria, if you mess with yeast, would it do something similar? Why not? Right? There's nothing special. Okay, so that was the thought experiment behind this project, and it was done by this undergraduate in the lab, well-defined project, 2 years, got to start and finish. Happily, it happened. So like I said, the question, to what extent long-term resistance is foreshadowed in early response to drugs, and to what extent the early response to drugs foreshadowing long-term resistance is evolutionarily conserved? Okay. So how do we do this? We collected primarily from these two papers, both from Arjun Raj's lab, that involve melanoma and breast cancer and vemurafenib. So they have published these long-term gene signatures. So we took a consensus of these 4 experiments, and we came up with a set of some number of genes. And this is our other independent validation data set, the same deal, but there is a time course data in another cell line, lung cancer cell line, treated with a different drug, and 0, 3, and 7 days. So we first built a consensus signature from those first 2 papers and tested their activity with time in the third the paper. Okay, as you see, it's a monotonic increase in the resistance as the time goes by, but it's there. So ultimately, we took these 3 consensus of all these data sets, involves 3 different cell lines, 2 different drugs, and put it together into our what we call quote unquote pan-cancer drug agnostic resistance signature. Okay? So that's our stand-in for resistance signature. Then what we do is go to a different data set, which is early response data set in single cell. So here it's a wonderful data set, 3 cell lines, 188 compounds, huge single cell data set. This is the number of cells for each of, and they cluster this gigantic set of cells in each of the cell lines. So this is just for A549 from lung adenocarcinoma. And this is the number of cells in each of the clusters they identify, blah, blah. Blue is the pre-treatment, and the red is the post-treatment, which is much bigger because it's combining 188 compounds. Okay? So that's the data. After several weeks, not the previous, our signature is based on long-term resistance. Now I'm going to map it to early resistance, early response in this data set. That's the idea. Okay, so first things first, we simply took our signature, long-term signature, and scored each one of these individual clusters in each of the 3 cell lines. So we see a huge variability in the score, and then based on some simple metric, we called specific clusters as RACs, resistance activated clusters, just some random name. Because they are enriched for these high scoring cells, high scoring for the long-term resistance. Okay. So we found these many racks in the 3 cell lines. And then we are really after something that is agnostic to the cancer type and drug and all that, right? So that's our goal. So we clustered all the individual racks, right? All these individual orange things across all 3 cell lines, we clustered them together using some technique that you can read up. Okay, so this is what it looks like. And it falls into 3 broad clusters, which sort of are pan-cancer, pan-cell line clusters. And first fact about these 3 clusters is this. If you score the cycling signatures, only cluster, we call them super cluster now. Super cluster 2 is cycling. The other 2 are sort of arrested, more or less. Okay. So again, this notion of variety of cycling states, cycling or non-cycling among the resistant cells has been noted before. So we are not the first one, but just confirming that that is the case in what we find as well. Okay, this is just also another, don't read it, just to show that in each of the supercluster, we looked at various enrichment of various gene signatures. And there's a lot of variety, right? This is the only one that is enriched for cell cycle. These 2 are not, and so on and so forth. Okay. Dr. Michael Gottesman, who happens to be a, synonymous to drug resistance as a, yeah, go ahead. No, only a small subset of these racks are pre-existing in the untreated cells. So we did that as well. But there are some, but not all. Some are indeed induced upon treatment.
Yeah. The signature could be because of both genetic and non-genetic factor, right?
Here we are completely assuming non-genetic. It's totally non-genetic. So where would genetics?
Be mutations which are causing this.
Right. Again, but that will assume clonality of some sort, right? This is all kinds of data set in all kinds of conditions and cell lines. you can't have, same mutations explaining all this variability. So that's the easy way to rule out genetics in this case. I'm sure there'll be mutations, but I don't think they're responsible for these very noticeable programmed concerted changes. Yeah, across cell lines, across treatments, everything. So Dr. Gottesman, he is very well known for this multi-drug resistance, ABC transporters. So he happened to have a whole bunch of whole genome CRISPR knockout data sets in his lab. So they are interested in knowing which gene knockout manipulates or modulates drug resistance to specific cell line, to specific drug. So he was very kind to just give us one of those data sets, this ovarian cancer cell line against this CHEK1/2 inhibitor. Okay, and the mechanism of action of this drug is replication catastrophe. So it's significant to keep that in mind because it will only kill the cycling cells, not the non-cycling cells. Okay, good. So this axis here, the higher the number, it means that knockout of that gene sensitizes, that sensitizes the cell to the drug, makes it less resistant. So we took our upregulated genes in supercluster 1, 2, and 3 and downregulated one and looked at its CRISPR knockout score. So you can see a similarity between cluster 1 and supercluster 3. And in both cases, knocking out, these are arrested cells, remember. So knocking out the genes that are specific to these superclusters are likely getting them back into cycling state. And that would be consistent with them becoming more sensitive to the drug. Whereas the supercluster 2, which is arrested to begin with, if you knock out the important genes in those, it might become arrested and it will become less sensitive to the drug. So that's also perfectly consistent with what we are seeing. Okay. And this is a standard. We have to always show clinical utility, blah, blah, blah. So this is we took the superclusters in K562, not RACs, and in leukemia, we show that it has a strong prognosis. Again, our superclusters are meant to be pan-cancer, agnostic to everything, blah, blah, blah. So we applied it to entire TCGA cohort, how does it track hazard ratio after controlling for whatever, sex and age and tumor purity and all that. And it's a weak signal, but it's consistent across all superclusters across cancers overall. Okay. This is interesting. First one, so RAC signature is a prognostic of relapse and progression, not just overall survival. So this is a very small data set, 8 patients with EGFR mutant lung cancer treated with tyrosine kinase inhibitor. So 4 short relapse, 4 long relapse, and we simply checked whether our supercluster signatures are, are higher for short relapse resistant signatures. That's the case. And this is another data set where we have pre-malignant lesions in breast, and some of them went on to become breast cancer, and some did not do in the observation period. And again, we asked whether the ones that went on to become cancer, whether they have, higher resistance. So again, which is the case, I'm taking something from resistance. What has that to do with pre-malignant lesions becoming cancer, right? It's totally unrelated context. So I don't know how to explain this, but this kind of carries, to these other contexts as well, this signature that we have. Okay, finally, looking at evolutionary conservation of what we are finding. So we went to these 2 data sets, E. coli and yeast. In case of yeast, there are acquired antifungal sensitivity and resistant genes. So against 9 antifungal drugs, again, we took a consensus of these genes, which were coming up in many of these antifungal drugs. And likewise for E. coli, 9 different classes of antibiotics, again, a consensus gene set. And we simply scored our single cell data, supercluster single cell data with these E. coli and yeast resistance signatures, okay? And in case of yeast, we see both supercluster 2 and 3 being much higher than other cells. And in case of E. coli, supercluster 2 is much higher than the non-RACs, non-resistant clusters. And these are specific processes and genes that are common. And here is those. So we're very happy to see these multi-drug transporter genes showing up in case of yeast. That's interesting. And so supercluster 2 is common to both of these. And these are the cycling ones, right? So seems like in yeast and E. coli, at least in E. coli, the prominent mode of resistance seems to be cycling, doing something, proliferating or something. So that's the end of story number 2. The take home is cell states in drug naive population as well as in population shortly after treatment share transcriptional properties of fully established resistance. And these signatures are prognostic and they're conserved. So that's story number 2. So we can take more questions. And before I move on, let your brain rest a little bit. Yeah.
This is really interesting what you're finding. But this is somewhat different from what Arjun's group has been claiming, right? From what I understand, from their slightly later papers, not the ones that you showed, right? What they are saying is that the so-called memory genes that we've been interested in also, right? Their signatures are there pre-drug. Very early post-drug, those also they find. But then in the long-term signatures, they find there is a lot of upregulation of completely new genes, which has nothing to do with these memory gene signatures. So in fact, they say that it's a different set of genes that completely get upregulated long-term, in long-term resistance. So I'm therefore very curious that you still find some signatures matching early versus long-term.
Right, right.
I wonder whether this is cancer specific or?
I don't know if it's cancer specific. So it could be that the fact that we are really talking about consensus gene sets across very different systems. So we are latching onto something really core, right? Whereas they are working on one specific system, in a single temporal data, you may see a lot of variability. But when you're talking about a lot of these may be just unnecessary, non-significant downstream effect of stuff. But the fact of taking consensus helps us get to the most important, truly causal one, quote unquote. And maybe in that sense, you may start seeing a greater overlap with the early memory genes, maybe. I do not know. Okay.
Can move to the next story.
Can we move on to the next story? Okay. And you stop at 1 o'clock, right? And that's in general, yeah. Okay, good. So I'll stop whenever clock says 1. Okay. Okay, I think I can at least finish 2 stories. Good. So next story is a little bit different. We are still talking about cell states. So stick to cell states idea. Okay. I'll change gear just to introduce some other concept, then I'll come back to cell states. So I don't know how many of you know this concept of synthetic lethality. So if you have, the general concept is you have 2 genes. If you knock out either one of the genes, then cell is still viable, but only when you knock out both, the cell dies. Okay? And that concept is relevant for cancer therapy. So you have breast cancer, BRCA mutation. In that background, if you do PARP inhibitors, because they both are targeting the same process, the repair, and the cell is much more vulnerable and dies. So that's called synthetic lethality. The word synthetic here really means not synthetic as in not natural, but synthetic as in synthesis. Okay, and there is another concept called synthetic rescue, which becomes relevant in resistance. So here is a, this BET inhibitors trying to turn down the MYC, but then the cell finds 2 alternative ways to bypass that and turn on the MYC anyway. So that's called synthetic rescue. Now, several years ago, we again asked this, because as a computer scientist, I look at something and I step back and say, is there anything special about it? Or is it a general phenomenon? That's the modus operandi of my research, basically. Okay, so we asked that. So I just laid down these two things. Synthetic lethality is this. So if you have gene X and gene Y, when both are low, then the cell dies. So that's the red. Here is the case when you have gene X, sorry, here let's say this is gene Y, which when it is low, like BET inhibitor, for example, then the cell dies. But in that background, if some other gene comes up, then cell is rescued, right? So you can represent synthetic lethality and synthetic rescue simply by these coloring this 3x3 matrix. Is that okay? Now, if you can do that, then what about all these other colorings? They're also viable gene-gene interaction situations. Okay? So this would be gene X, when gene X is low and gene Y is high, then the cell dies. But when gene X is high and Y is low, then the cell is extra proliferative. So that's the way I would read this. Okay, so back in the day, we kind of did this. We identified all kinds of gene interactions and showed that synthetic lethality, although it's there, it's a minority of all types of gene interactions that exist in nature. Why we focus on synthetic lethality? Because it provides us a very clear-cut way of therapy. Because you have BRCA mutation, you do PARP inhibitor. So that's the reason SL is the most popular one. But if you talk about the cellular biology, all these other things are all real. Okay, now I'm going to move on to from genes, I'm going to move on to something called cell state that you already know what it is. And it makes sense to talk about cell states because genes don't work by themselves, they work together, and together cell is the ultimate unit of function. Even that some may argue because cells have to work together in the tissue, different types of cells. There's a whole mess in the tumor microenvironment, right? So for example, if you have more mesenchymal cells, then it's more resistant to therapies and more worse prognosis, so on and so forth. So that's the notion of cell state that you might recognize. Okay, so we did the following. We are doing this, it's not a done work. So we literally translated this notion of gene-gene interaction. I'm just showing this for synthetic lethality to cell state interaction. Is that clear? So you have malignant cell state A, T cell state B, and only when both states are present, you have good prognosis, otherwise you have bad prognosis. So that's what I mean by interaction. Not interaction in terms of physical interaction, not interaction in terms of co-occurrence, interaction in terms of when they co-occur, it has a very specific outcome, clinical outcome. So what do we need to do this project? If we had this data, single cell RNA-seq data with clinical outcome, then from single cell RNA-seq data, you could derive cell states, which is not hard, easy. And then you could simply, for each pair of cell states, you could ask whether it correlates with specific clinical outcome or not, right? So that would be the obvious thing to do. But this doesn't exist. Single cell data with large cohort of single cell data with clinical information does not exist. What we do have is TCGA type of thing, right? We have bulk data with clinical information. So if only we could take the bulk data and from that derive single cell states of each cell type, then we would be in business. Okay? So that's what we do. And how do we do that? The idea is this. There is a thing called deconvolution. I mean, some of you might have heard of it. So the idea is from the bulk data, can you get cell type specific gene expression out? So the way it works is, I mean, you know what type of cells are in that mix, and you know what their signatures are. So you use those signatures to statistically deconvolve this bulk data, and you get data for each cell type in each sample. So that's how deconvolution works. And once you have the cell type specific data, it's almost like cell sorting. In the bulk sample, you're sorting T cell, you're sorting epithelial cell, and you're doing sequencing. That's what it basically boils down to. If you have that data, then you can do some latent factorization for, let's say, epithelial cells across samples. You can factorize it to get states. Right? So when I say factorize, you may have heard of PCs, of course, principal component. You may have heard of non-negative matrix factorization. That's another way of factorizing. But what we chose is something in between. It's called independent component analysis. I won't get into the technicality of it, but it suits our needs much better than NMF or PC. Okay? It really tries to capture cell state better. So that's what we do. This is a huge pipeline, TCGA. We have singles. All this is applied to IDH mutant glioblastoma, by the way. So everything is IDH mutant glioblastoma. So we have single cell data that we use to deconvolve, and from that we apply our ICA to get cell states, and then we do the 3x3 matrix business to see which cell state combination track clinical outcome. Okay, and so that's what this is basically. Hazard is equated to state 1 combination while controlling for individual states and age, sex, and purity. As simple as that. Okay, so we apply it to, like I said, IDH mutant glioma, and we chose to use 10 independent components per cell type. So that's our guess. We have reasons to choose that number, but that's kind of maximum 10 cell states per cell type. And these are the total number of pairs that we test. So independent components is a statistical entity. It's a combination of genes, right? That's what it is, just like PC, just like NMF factors. But what is the physical interpretation of an IC? We are hoping that an IC corresponds to a cell state. How do we know that? For something to be a cell state, in the single cell data, it should cluster. It should fall near each other. If it is all over the place, then it's not a cell state, right? So our ICs were designed without anything in mind, but we are asking how many of our ICs are actually a cell state, i.e., how many of them cluster in the single cell data, right? 70% do. So these are just 2 examples, right? So here is IC number 6 is some number, and we map it on the single cell data. You can see it nicely clusters. IC1 in T cell, they cluster, IC6 and so on. So we do our magic, we get 160 interactions, okay? Cell state of cell type 1, cell state of cell type 2. When they co-occur or when they are like this or like this, then you have worse prognosis or better prognosis, that kind of stuff. And first things first, majority of our interactions are pro-tumor. So that combination actually corresponds to worse prognosis, pro-tumor, worse prognosis. That makes sense because what we are seeing is a result of selection. So that's good. This is just to show that when we take whatever we learn from TCGA and go to an independent data set from China called CCGA, it exactly holds up. Okay, so it's robust. Whatever we're finding is robust, at least in terms of independent cohorts. This is the type of stuff we do as a computational biologist. Now the issue is challenge of interpretation. We have all these beautiful ideas. We can implement it. We can come up with 160 interactions. What does it mean, really, right? That's the bottom line. And that's what we struggle. And that's where the meat of our biological thinking comes in. So how do we go about interpreting? Focusing on malignant cells and see what their ICs look like biologically. Do malignant cell ICs correspond to known pan-cancer inter-tumor heterogeneity programs that have been published before? So give you an example. So this, IC number 6 of malignant cell, it cleanly corresponds to previously known astrocytic cells. Dimension 5, IC5 cleanly corresponds to this oligodendrocyte progenitor cells. IC7 cleanly corresponds to something called cycling cells. Now, glioblastoma stem cells are known to be cycling. So then the question comes, is this IC7 capturing stem cells? Glioblastoma stem cells, right? So we do additional analysis to figure that out. I'm just showing that these things cluster, so they are indeed. So first thing we do, we go to an older data where they show the differentiation of cells in the glioma, and our cells indeed map here, which is the progenitor cycling cells. We explicitly make gene signatures of putative stem cell signature from various sources, and again show that our IC7 cleanly maps to those. We also check embryonic and fetal and post-birth human brain data set, and we show that IC7 has the highest signature in embryonic and goes down in the adult brain. So there are multiple lines of evidence for us to believe that IC7 is indeed likely IDH mutant glioma stem cells. Okay? Just to show you an example of how we go about interpreting things. This is a good example. We don't get this lucky more often. So now what does it interact with? This is IC7, like I said. These are all its interactions. I'm going to focus on one of them, which is yellow T cell, IC1 of T cell. Now, what is IC1 of T cell? Again, we depend on the prior knowledge to interpret these things. IC1 of T cell happens to be senescence T cells, senescent T cells. Okay? So what does it mean? Stem cell, malignant stem cell, when they co-occur with senescent T cell, is going to do something weird. That's what this means. Okay? And that's what is illustrated here. So stem cell by itself has some prognostic power. Senescent T cells have some prognostic power. When you combine them, it has much greater prognostic power, right? The colors are interchanged here, I think. Okay. Now, again, the question comes, if two things are interacting, like I told you earlier, our definition of interaction doesn't mean physical interaction. It doesn't mean co-occurrence. It means when they occur in the same tissue microenvironment, then they have a correlation with outcome. But to what extent some of these interactions we are detecting are actual physical interactions? It doesn't have to be physical like this. It can be secretion of cytokines which have limited, geographic range, whatever. So that would also make it physical, proximity-based thing. So that's what we checked. It checks out. In general, we do ligand receptor proximity analysis in the spatial transcriptomic data sets, 6 of them, and in general we see that our detected ones which are supported by ligand receptor pairs are very enriched among the ones which are spatially proximal. So a subset of our interactions are indeed mediated by ligand receptor type of interactions, but not all. And this is just an example showing that, so the red dots here are where both of these states are in the same Visium spot, so co-occurrence. And a lot of red spots here. And this happens to be between tip-like endothelial cells and EMT, the malignant stressed EMT cells, okay? EMT stress cells. And again, the same kind of deal. This is the last bit in this story. It's super interesting. We don't know what to do with it. This is just showing that our existence of our interacting pairs in a sample can help us predict something, right? So there's only one data set in IDH mutant glioma, anti-PD-1 therapy, 15 non-responders and 12 responders, and it adds up. The pro-tumor are enriched among the non-responders, and the anti-tumor CSIs are enriched among the responders. But this is just saying that you can do much more with our CSIs in terms of classification of IDH mutant gliomas. Currently, it's just A type or O type, but when you score them, a subset of O and subset of A start to look similar in terms of their survival. So we're trying to refine the clinically followed classification of these cancers. Okay, this is the last bit I mentioned. Somatic mutations can alter the tumor microenvironment. Makes sense. Okay? So can somatic mutations also alter our interactions, the cell state interactions? That's the question we were asking. So don't have to read this plot at all. All we did was in the TCGA, we had the mutation information for all the major oncogenes. For each Actually, we did for every mutation that occurred in at least X% of patients, basically. So just to have enough statistical power. And we are correlating the mutations with the existence of a specific CSI, cell-state interaction pair. Okay? So all these are either positively or negatively correlated. These are the genes that are mutated, and these are the cell-state interactions. Don't worry about it. But this is what we saw. Overall, as I told you, majority of our CSIs are pro-tumor, as one would expect, because we are talking about tumor that has gone through selection. But we only look at the ones that are correlated with mutations. Those are predominantly anti-tumor, which is totally crazy. Okay? So that gets your mind going, like what's going on? Is it like a homeostatic response to mutations? Right? If you go with that theory, we are imagining something like this. So early stages of tumor, you have more homeostatic, like anti-tumor interactions here that are taken over, other things overwhelm it, and it becomes more pro-tumor later on. So we simply tested this hypothesis. If this is true, then stage 1 versus stage 4, stage 1 and 2 versus stage 3 and 4, will we see more pro-tumor guys happening later and more anti-tumor guys happening earlier in terms of, and that's indeed the case. So that's all the evidence we have. And then based on this observation, we started a new project, spent 3 months on it, and we have abandoned it because we cannot come up with a convincing way of defining what's a homeostatic response is. If any of you have any idea how to capture it, how to quantify this notion, I would love to talk to you, but we could not. So we thought of many different things, and we hit a wall. It's like it's trivially explained by this, so forget about it, that sort of stuff. We are nowhere with this, but that's the observation we have. Cool. So that's the story number 3. What is it, 4? I don't know. 3. Any questions before I tell you one more story, not 2? It was clear? Just give me some nods at least. Yeah. Of course, that's fine. What do you mean by suppression of what? Anti-tumor. Yeah, exactly. Oh, no, we find both. We find both. That's what I call pro-tumor and anti-tumor. Right. Mostly they're pro-tumor because we're talking about cancer, but the ones that are associated with mutations that are largely anti-tumor. By themselves, they predict better prognosis. Yeah. Sorry, you're too far.
I don't want to take too much of your time because I guess you want to finish, but maybe just to bring in something and we can continue later. But I'm imagining that if you have a mutation, you probably get a response from the environment. So, and this would happen presumably on a longer time scale than the more sort of protomorigenic events which occur between interactions that occur between, I don't know, angiogenetic factors.
Right.
So I guess if I think about it, maybe, of course, with hindsight, it seems that I would actually, in fact, expect mostly anti-tumor interactions with respect to mutations because the mutation appears in a kind of a wrong cell, and then the body will try to, it's a bit like, I'm just thinking if it has something to do with like, you probably know the HIV, anti-HIV response, right? That you get this incredibly high turnover of T cells trying to respond to this over a very long period of time.
Right.
Until they
yeah. So multiple things, right? We need a data, like time data, like when the mutation happened, if we can figure out like what happened right after, right? That's one thing. Ultimately, we see all these changes, downstream changes that are associated with mutation. Which of these are downstream changes that are neutral? They're just there. which one of them are actually helping like grow, which ones were like relics of anti-tumor response, homeostatic response at some point, right, that are not functional anymore. So we went through all this thought process. And another factoid, in general, the oncogenic cells with oncogenic mutations in a tissue are typically outcompeted by wild type cells. So again, we think of oncogenic mutations as an all-powerful thing, but they're not, they're weak generally. Lots of things have to fall in place for them to proliferate eventually. And the third thing I would say is that I imagine these things to be very local. You have mutations, some local tissue changes, in the ECM, in the environment, and not so much systemic, but who knows? I don't know enough. Yeah. There is no way to quantify long distance interactions in spatial transcriptomics because oh, yeah, lots of them. Only a subset of all the things we detect are spatially proximal. They're enriched, but many of them, most of them are not explained by spatial proximity. Exactly. I don't know enough to interpret all those details, right? That we generate all this stuff, hypothesis, and we do at the level of high level pattern stuff, we do some validation, but to really get into interpreting these individual interactions, yeah. Yeah. No, we do log of hazard. We just, I do log of hazard. Right.
Question is,
What is that's a good question. I plead ignorance. There are armies of biostatisticians who work on hazard ratio models, and I don't know enough. I just use what's the standard in the field. Yeah, good question. I don't know. Okay, last story very quickly. So there were two parts of the last story. One of them I talked at IISc. I will do the other one right now. Okay, so that everybody got something fresh. So this has to do with I'm moving away from transcription as a first degree thing, but I'm moving to transcription as a second degree thing, mediated by single nucleotide changes in the genome. Okay. So this is a story, right? When you talk about mutations or polymorphisms, 90+% of them fall in non-coding regions, and we don't know what to do with it because we don't know what to do with it. Yeah. And why don't we know? Because there is no good model, right? There's no good model to say if you have a nucleotide A that becomes a C, what will it do to the regulatory function of that piece of DNA? That's the question. If you could figure that out, then you could start interpreting all this non-coding stuff in the genome. So that's what we are trying to do. This is just an example. I'll skip it. There are many examples. So these questions can be studied at 3 different levels: at evolutionary level, right? What happened during species evolution? What happened during population divergence, polymorphisms, how that affect our disease propensity from the non-coding SNP perspective, and finally somatic mutations in cancer, which also happen, most of them in non-coding region. Are they totally useless or neutral or what? Right? I don't think so. So the question we first question we went after was trying to figure out: are there during human lineage, in the human lineage, since macaque, are there single nucleotide mutations that potentially might have created new enhancers in embryonic brain? Okay, that's the question. Hopefully, finding some piece of the puzzle of human cognition. That's a bigger picture. So the data is from this Riley et al. Science paper in 3 species at different embryonic developmental time point. They did a bunch of profiling. Okay, one of them is K27AC. K27AC, if you don't know, it marks active enhancers in a cell. So that's our hero here. And we only care about human and rhesus here. Okay, so that's the data at specific equivalent time point. Okay, And just something about human cortical development is uniquely characterized by these facts: increased duration of neurogenesis, increased number of diversity of progenitors, neuronal migration, and new connections among functional area, interneurons. So this becomes relevant when we try to interpret what we find. So that's why I put it up here. So, so this is the aim. So like I said, if you change A to a C in a non-coding region, what does it do? So I have worked on transcriptional regulation field for a long time with very model specific thing. You have a TF binding, this transcription factor, its expression, and this other cofactor, put it everything together and try to predict the, but all that is highly limited and based on lots of assumptions, incomplete information. So this is a situation when we turn to deep learning. Because I know whatever is happening in this region, 1 KB, 2 KB, 5 KB, whatever, and I don't understand how it works, but the machine can figure it out. So what we have is a machine that, given a piece of DNA, it outputs its enhancer potential in that context. Okay? Higher this number, more likely that piece of sequence is to work as an enhancer. So we trained this, and this is just showing the accuracy of the model itself. I will move on. But here is the beauty. Our model is trained just to classify enhancers from non-enhancers, right? It doesn't know anything about A to C, C to G, none of that business. It doesn't know anything about it. And yet, when we have that information in a RAQTL type of experiment where people have changed one nucleotide and measured the activity of the enhancers, the gene reporter assays, right? Anytime our model says that allele B is more active, how often it's correct based on experiment, right? So that's what this is trying to show. As soon as the difference of the score, the sigma score between the two alleles, as soon as it reaches about 0.1, we are 100% correct in terms of getting the right allele as having higher enhancer activity. So this is wonderful. It came out as a side effect of this model, because model was not told anything about which nucleotide is important and all that. It just knows about this 1 KB or 2 KB sequence. So this is unbelievable to me, but it works. So we have that K27AC data, as you know. So that's what, so let's say you have K27AC peaks in human, macaque. And if you have something here and not here, we don't know whether it's gained in human or lost in mouse. So we need an outlier group, right? So outlier, but we don't have the K27AC data for the outlier. It doesn't exist. So that's when we go to our model. So our definition of a gain is something has to be here, not here, and our model should say yes, our model should say no, and our model should say no. And we infer this ancestral species sequence based on probabilistic consideration, like phylogeny, tree building, that type of thing. So that's that's how we ascertain what is gained, what is lost, and so on and so forth. Okay, so we apply our model to all the mutations that we detected in human lineage that fell in one of these cases. Okay, sorry, right now there's no mutation. I apologize. So this is just about the enhancer gain and loss. So we detected that many gained and lost enhancers, and did some tests. So gained enhancers have higher expression in human compared to macaque. These gained enhancers are more enriched for the EQTLs in brain, which is nice, not the lost ones because they don't work in human. They are enriched, the genes near them, well, we did a great analysis. The genes near them are enriched for all these processes. And this is nice because this sort of maps to the things that are unique about human cognition development, or brain development rather. At the flip of the coin is that the central nervous system disorders, GWAS signals, are also enriched in these gained enhancers. Again, this is a two-edged sword, you know, a good and the bad with everything. We were interested in knowing like what specific cell types in the brain these enhancers are active in. Okay, that's an important question. We relied on the single cell data in embryonic brain, human brain. And for each cell type, we quantify the enrichment of the activity of the enhancer in that cell type compared to other cell types. And the ones in bold are the ones that are enriched. And if you look at their names, progenitor, interneuron, cycling progenitor. So basically progenitors and interneurons just light up with these enhancers. There's no reason for it to happen. Right? There's no bias in the analysis. Yeah. I haven't come to mutations yet. This is all about the gained enhancers. I'll come to mutation. Yes, that's what I'm saying. Correct. Yeah. I will come to that. Yeah, absolutely. So now we're coming to the mutation. Now, in an enhancer region between human and macaque, there will be lots of mutations. 2, 3, 4, 5, 10, who knows, right? Now we ask, okay, you start with human. I change one nucleotide to the macaque version. How does it change the enhancer score? Is it large enough? Then I change the second one at a time. Then I go to 2, each pair, okay, like that. So this is like in silico mutation to see which are the critical mutations, which change the enhancer score sufficiently, right? Make something from inactive to active based on the enhancer model. So we do that. And turns out that about 40% of these gained enhancers, one mutation is sufficient to change it from inactive to active, based on our thresholds, of course. Everything is threshold. 40%. Others require more than one mutations, according to this state. Now, we just focus on these, right? Because we are sure in this case, like one mutation, which one it is, and what it does. We call this essential mutations, and I'll show you some data on the essential mutations compared to non-essential mutations. So what's special about essential mutations? They show higher allelic imbalance. So you have 2 alleles in a genome, right? So, and if you have heterozygous site, the allele that our model says has higher enhancer activity should have greater K27AC reads compared to the other allele in the same cell, right? So that's what allelic imbalance measures. So there is a way to measure allelic imbalance at heterozygous sites. Of course, they're limited to this test only on heterozygous sites in that sample. So that's true. Essentials are more heterozygous. Essential mutations are more likely to lead to expression increase compared to non-essential ones. Essential show greater enrichment of cognitive trait signals, the one I showed before. So whatever I did before, these things are even more along the same line. So they're more enriched for all these things. And then I come to this transcription factor business. So what are these essential mutations disrupting, right? So we do our standard motif, like which motif is being disrupted at these sites specifically. And these are the guys. So all these are increased activity, all these are decreased activity. Okay. And then we do a very sophisticated sort of regulatory network building for these transcription factors to come up with a core regulatory network. And it has directionality. I won't get into that, how we get the direction, but POU3F2, it sits on the top, and all these other subordinate transcription factors, and these at the end. So what we are saying is there is a set of mutations that happen in the entire genome that recruited and changed the regulatory network in a way that was conducive to specific aspects of embryonic brain development in human, different from macaques. So that's it, my second story. In a nutshell, this one, I will just tell you the question. I won't show you any results here. Question is inspired by this fact that African Americans have much higher risk of prostate cancer, and polymorphic germline susceptibility is one of the major causes of cancer in general. So again, we ask the same question: what's special about polymorphisms that are high frequency in Africans that might affect prostate enhancers, that might affect susceptibility to prostate cancer, right? That's the idea. And just to honor our friend Dimple here, I'll just tell you that this project was inspired by this one slide, literally. So where this particular variant has been associated with twofold increase in prostate cancer risk. And Dimple's lab showed that this, in the normal allele, this SPDEF binds kind of weakly, and it has a weak 3D environment around it. But the risk allele really strengthens this binding and turns up a bunch of link RNAs and MYC in that locus. And that kind of functionally links this variant to prostate cancer. And we said, okay, so this is our poster child, and we do it in a genome-wide fashion. In the process, detect this and a bunch of other things, and what those things, other things might tell us. So that's what we did in this project. I'll tell you one factoid, very cool, very, very cool. So if we identify all these SNPs, right, that have high frequencies, African Americans, and where the allele B, which is higher in Africans, increase the enhancer activity. And then we asked, what are the genes functions that enrichment for these genes, these types of genes? Of course, you see lots of things, but one of the things that came up was this regulation of telomerase activity. That's super interesting. What would that imply? Would that imply that African cells derived from Africans have higher, longer telomere? So we started talking to lots of telomere experts. Can somebody do the experiment to take 2 cell lines, white derived, African American derived, and measure the telomere length? And of course, in the meantime, we found this paper published, and that's what they have done in different tissue, different cell lines of different tissues. They have measured whites versus blacks telomere length specifically. And guess what? Prostate is on the leftmost end here, right? So this is completely agnostic to anything, and that kind of reveals this fact that was already out there, which we're happy, and some other things. Yeah. That's it. I didn't want to stand between you and your lunch. More questions?
Related to your human versus macaque story.
Yeah.
Did you come across FOXP2, which is responsible for speech?
Yeah, of speech, lots of foxes come, lots of foxes. Many foxes. Look, it's chock full of FOXP1 is there too, right there.
This human versus macaque?
Yeah, FOXP1 in both gained and lost. But then there's so many foxes, and foxes have such so similar motif.
FOXP2 is highly correlated with speech. Like
Yeah.
any mutations in FOXP2 speech.
Well,
effects.
yeah, right. But we are talking about binding sites, not the protein
Because
itself.
that is the striking difference between macaque and
Yeah.
speech.
FOXP1, but again, like I said, the motifs can be ambiguous. It can be FOXP2 motif, we don't know, right? Yeah, I don't even know if in the database there is a specific motif for FOXP2 or not. Because if there is none, then we will not even look at it. Yeah.
Yes.
Yeah, can you please, because I'm too far.
So whatever essential mutations you have found, so is there any time limit, time points like this mutation happened fast, this happened later, and that's why the enhancer activity increased, because in number of, whenever she's seeing in the 2, maybe 1 has to happen first, then it has to happen. That's why in the 2 it is slow and it's getting lower and lower.
That's a good question, but ultimately all our mutations are ascertained based on species genome comparison. All we know that it happened since the common ancestor of macaque and human in the human lineage. That's all we know. Where in that line it happened, it's impossible.
One more question, that you have also seen 3,000 genes they have decreased enhancer.
Yeah.
What are your thought on those? Is there any specific motives or?
Right. I mean, the thing is, based on our initial analysis, a lot of signals were absent from the lost ones. So we pursued the gained ones only in that project. Yeah.
The lost ones doesn't have any specific features?
Right. Didn't find any enrichment or anything. Why? Yeah. Yeah.
There has to be a reason. I mean, it's
Yeah.
yeah.
What was the other reason why they have these
It's a
alleles
big
and that has
disadvantage, yeah.
been hijacked for some other, yeah, yeah. True. These are deep questions. I need help. And
This might be true for other cancers also, in African ancestry or even the metabolic disorders.
right. Yeah. I don't know what other cancer types we know, like clear cut, such a big difference in incidence rates between populations. Breast cancer also? Okay.
Skin cancers, they're less susceptible. African American. Yeah. Right.
Yeah.
So African Americans are protected from melanoma because of
But
the,
that could be more physical
Right,
reason, more
yeah.
physical. Yeah. Liquid tumors. Yeah, liquid is going all over the place, but right. Right. Yeah, I mean, so we are talking about healthy cells here, remember, right? So what does it mean in that context? If you take blood, right, or WBC, whatever, blood. So blood is all over it's coming from somewhere in the body, and then you look at heterogeneity. Of course, you'll find all kinds of T cells in there. Then we are talking about within each T cell. Yeah. Okay. I haven't. Yeah. No, exactly. The RBC is not a good candidate. Like B cells might be. B cells might be a good candidate. Yeah, we haven't.
I'm just curious if there's like big differences between solid and liquid tumors in terms of
Right.
like when it becomes.
Okay.
more questions. Let's thank Sridhar for the wonderful talk.